morning, good afternoon, and good evening. Welcome back to Chapter 4. We're talking about cells. Now we're talking about the organelles. We're talking about the little structures inside of a cell that have particular jobs, particular functions that they do. And our first organelle is the nucleus. The job of the nucleus is to contain the DNA, to be essentially the library of blueprints for the cell. Blueprints on how to make all of the features, all of the structures, all the molecules that a cell needs to do its job. I want you to imagine that a cell is like a little miniature factory. It is the nucleus that holds the recipes for all the chemicals the factory needs to make. When that DNA is inside of the nucleus, it's in the form of what we call chromatin. And I know when you think of a chromosome, when you think of DNA, you probably think of that X shape. But that X shape is only present during mitosis. You know that mitosis is cell division. Cell division occurs at the end of a cell's life. When it's done, it splits in half and becomes two cells. Outside of that cell division event, DNA is not in that X shape. That X shape is like if you took a big pile of yarn, not of yarn balls, but just a big pile of yarn. Imagine you take all the yarn and you unwind it all, and it's just a big clump in the floor. That is what DNA is like inside of the nucleus. It's just a big clump. That X shape is if you took all of the separate strands of the yarn and wrapped them back up in their individual yarn balls. That's what the X shape is. It has been wound up, it's been wrapped up, twisted up to make it easy to separate, to make it easy to handle. And when it's not in that chromosome shape, that X shape, it's in this unwound shape, and we call it chromatin. Chromatin is when the cell is not dividing, it's just the DNA inside the nucleus. It's what you call the DNA what's inside the nucleus, chromatin. As we said before, it's the job of the nucleus to control all the cellular activities, and this is where RNA is made. Messenger RNA is made inside of the nucleus. We already said before that the nucleus is surrounded by a wrapper, and that wrapper is called the nuclear envelope. This envelope, as we said before, has holes in it, and those holes are called pores, nuclear pores. And the pores allow for things to go into and come out of the cell. To go into and come out of the nucleus. Specifically stuff like that messenger RNA that we just talked about. The messenger RNA is a copy of a DNA recipe. And the messenger RNA, I kind of imagine it like a sea snake, like a ribbon swimming, and it has to swim out of the nucleus, and it does so by going out of these pores. The nuclear envelope is continuous with the rough ER, and we said that in the last video. What does continuous mean? Continuous means that it's hard to tell where the one stops and the next one starts. So the nuclear envelope is continuous with the rough ER. One last structure that we have within the nucleus is called the nucleolus. The nucleolus does not have a wrapper of its own around it. It's just a darkly staining region of the nucleus where ribosomes are assembled. The nucleolus is a darkly staining region of the nucleus where ribosomes are put together. We talked a little bit about ribosomes. We'll talk more about them in a few minutes. So here is our stereotypical animal cell. And this is specifically highlighting the nucleus. So here's my nucleus. The nucleus is bound. It has a wrapper around it called the nuclear envelope. That nuclear envelope has little holes in it. Those holes are called pores. It's the job of the nucleus to hold on to the DNA. The DNA, when it's inside the nucleus, is all unwound. And so it's just a whole bunch of strings. And when it's in this string form, it's called chromatin. There's a darkly staining region of the nucleus called the nucleolus. And this is where ribosomes are assembled. And our last piece of this is that the nuclear envelope is continuous with the rough ER. And here we see the nuclear envelope flowing right into the rough ER. One last thing I want to talk about before we go on, and that is why the rough ER is called rough. Do you see how it's got ribosomes stuck to it? Kind of looks like it's studded, it's pimpled with ribosomes. That's why it's called rough, as though it would be rough to the touch. Obviously, it's not rough to the touch because you can't touch ER, too small. But that's why it's called rough ER, because it's got little ribosomes stuck all over it. The job of those ribosomes is to take the directions from the nucleus and follow those directions and make the product that the directions tell the ribosome how to make. Those directions are copies of the original blueprints, and those copies are called messenger RNA, mRNA. And it's not a whole copy of the whole set of blueprints. It's just a copy of a single gene, of a single product, set of instructions for a single product. It is the job of the ribosome to assemble the amino acids in the right order. And we talked about this when we talked about proteins in Chapter 2. We said that it is that amino acid sequence, primary structure, that is what determines the overall shape of a protein. And it is the ribosome that puts the amino acids together in the right order by following the instructions on messenger RNA. Ribosomes can either be free or bound. Free means free floating in the cell, or they can be stuck to the rough ER like we saw in that previous picture. And it doesn't have to be an all or nothing principle. Sometimes they can be bound if the particular protein they're making needs to be released by the cell. And other times they can be free floating if the particular protein they're making does not have to be released by the cell. So it could be free floating 1 minute and then be bound a minute later. They can go back and forth. So here we see again my animal cell. Here's my nucleus, and we said that the rough ER is continuous with the nucleus. Here is the endoplasmic reticulum, the rough ER, and we see there are ribosomes stuck to the rough ER. Here is a breakdown, here is a blow-up of a ribosome, and we can see that piece of messenger RNA, that set of instructions running through the center of it. And we see this growing protein. Remember the little pearls that we talked about? Each one of these little dots, each one of these little pearls is an amino acid. It's the job of the ribosome to string together the amino acids in the right order to make a particular protein. If a ribosome is specifically stuck to the rough ER, it's stuck at a hole because the rough ER has holes in it. And if a ribosome is stuck to the rough ER, it's stuck at one of those holes because this protein has to be released outside. And so when the ribosome is making that protein, it is feeding that string of pearls down into the rough ER, down into one of the holes that we see in the rough ER, so that it can be inside of the rough ER, so the rough ER can do its job, which is to modify that protein that's being made. If a ribosome is stuck to the rough ER, it's stuck at one of these holes. The hole allows the ribosome to feed the growing protein that it's making, that it's creating, down into the rough ER. The endoplasmic reticulum. What does endo mean? Endo means inside. Plasm, we'll take that to mean the plasma membrane. The plasma membrane is the outer skin of a cell. So inside the skin of the cell, inside the cell. Reticulum. Retic or reticule means folded. So endoplasmic reticulum literally means the folded stuff inside the cell. What is the endoplasmic reticulum? It's the folded stuff inside the cell. It's a bunch of folded membrane inside the cell. Extensive network of flattened sacs and hollow tubes. The ER, the endoplasmic reticulum, comes in two forms. It's either rough or smooth. We've talked quite a little bit about the rough. The rough is a series of interconnected flattened sacs. It's called rough because it's got ribosomes stuck on the outside of it. The rough ER makes proteins that are specifically supposed to be secreted outside. The smooth ER detoxifies proteins. The rough ER is a bunch of hollow flattened sacs. The smooth ER is a bunch of hollow tubes. The rough ER is continuous with the nuclear envelope. The smooth ER is continuous with the rough ER.